this is, you can see a lymphocyte treated by ML1, and it shows the typical apoptotic bodies. Another mechanism of mistletoe lectin-induced apoptosis is indirect. ML1 or mistletoe lectins induce the so-called Fas ligand on lymphocytes. And when these Fas ligand expressing lymphocytes bind to tumor cells that express the Fas receptor, the tumor cell undergo apoptosis. And this effect may explain how tumor cells could be affected by activated lymphocytes after a subcutaneous application of Viscum album extract. Now, viscotoxins, another important compound of mistletoe extracts, it mainly induces necrotic accidental cell death. This is not the physiologic cell death. Viscotoxins are small amphiphilic basic peptides, and their cytotoxicity depends on viscotoxin fraction. This is actually a fraction of different proteins and on the cell line. Viscotoxins rapidly permeabilize the cell membrane. They generate reactive oxygen intermediates, a swelling of mitochondria, and degradation of cytoplasma and chromatin. And here you can see a lymphocyte that has been treated by viscotoxins, and the cell membrane is lysed on the right side. Most investigations deal with immune modulating properties of mistletoe extracts. They also depend on host tree preparation and compound. Monocytes, macrophages, NK cells, T lymphocytes are activated. Their cytotoxicity, phagocytosis, antigen presenting function, cytokine release, and their locomotory activity is increased. And very interesting, the adaptive application of mistletoe activated macrophages or mistletoe activated NK cells inhibit tumor growth and inhibit metastasis in mice, just giving the activated cells without any mistletoe extract. Very interesting, the polysaccharides, so-called rhamnogalacturanan. There were many experiments by Max Planck Institutes, and they bridge tumor cells and effector cells, different effector cells. They have a bridging effect, and by this bridging, they strongly enhance the cytotoxicity and the sensitivity of the tumor cells. Granulocytes are also activated, especially by viscotoxins. A variety of cytokines is induced, especially pro-inflammatory cytokines. They have adjuvant effects. That means they augment the humoral and cellular immune response to other antigens. And they also have some other effects. I don't want to go into everything here. Two different research groups have found DNA stabilizing properties of mistletoe extracts. For instance, DNA repair is improved in cancer patients in vitro and in vivo. And here you see the DNA repair of breast cancer patients while they are receiving mistletoe every day. A possible implication might lie in reduction of side effects of chemotherapy and radiotherapy by mistletoe therapy in cancer patients. And I'll show you some studies on that later. Also, the sister chromatid exchange, this is an indicator, a sensitive indicator for DNA damage, is reduced in molecular cells. This means DNA stabilization too. The spontaneous and the cyclophosphamide-induced DNA lesions and the reduction of activation marker expression by cyclophosphamide is reduced by mistletoe extracts in vitro. But this does not necessarily mean that it leads to an inhibition of the cytotoxic effect of cyclophosphamide. On the right, you see an experiment where animals were injected with cancer cells, and they were treated with cyclophosphamide and or with viscotoxins, with mistletoe extracts. And cyclophosphamide reduced the number of metastases, and the mistletoe extract did so too, and both together had a stronger antimetastatic effect. Anti-tumor activity was investigated in about 100 animal experiments, and there were a variety of endpoints were investigated, like increase of survival, inhibition of tumor growth, tumor regression, reduction of metastasis, etc. These are far too many just to present here. On the right is one experiment which shows again anti-metastatic effects of mistletoe application with lymphosarcoma cells that were injected in mice. Very interesting is that oral application were also effective. Usually you give mistletoe extracts subcutaneously or intratumoral, or in these experiments also intraperitoneal. On the left you can see the animal models that responded, and on the right you can see the animal models that did not respond, or the tumor models that did not respond to this therapy. Now I will go to clinical trials. We have conducted 2 major systematic reviews on mistletoe therapy in cancer to clinical trials. The first we published in 2003, and this was on all mistletoe extracts. Now we have conducted a new systematic review which was just on anthroposophic remedies, which are far the most and widespread and most investigated mistletoe extracts. And this new systematic review was part of an HTA report in anthroposophic medicine, and it had the preset aim to include also very naturalistic investigations with high external validity, so we also included retrospective trials. But what we found that irrespective of methodological considerations, the quality of retrospective trials was just far lower than the quality of prospective trials. Especially the retrospective comparative trials have a very low quality and often have a lot of biases. And therefore, for the following presentation, I will concentrate on prospective trials, on randomized controlled trials called RCT, or non-randomized controlled trials, and RCTs and cohort studies, just on anthroposophical mistletoe remedies. We searched 9 electronic databases, reference lists, experts, and we contacted experts and manufacturers. We had certain selection criteria. We only collected trials on clinically relevant outcomes. That means not just on immune modulation or on toxicity. And we also included unpublished trials. And then we conducted a criteria-based quality assessment of the trials. This was the result of our literature search. It was quite adventurous. I don't want to go into the details, but this lists what we found initially and what we excluded and why we excluded it, and what we finally included now for the prospective trials. Here I can give you another overview on the mistletoe trials, mistletoe in oncology. 91 trials, clinical trials we have found, 22 prospective controlled trials. Of these, 14 were randomized and 2 were double-blind. 9 prospective cohort studies. And then, what I'm not going to show you, 23 retrospective cohort studies. Among these are also the cohort studies that were not clearly claimed to be prospective, and 37 retrospective controlled trials. 2 of them are retroactive, and these 2 have a lot better quality than the other ones, and I'm shortly going into one of these later.